This video describes the combinatorial synthesis of biodegradable polyanhydride film and nanoparticle libraries, and the high-throughput detection of protein release from these libraries. First, a high-throughput automated deposition apparatus is used to deposit a compositionally diverse array of monomers into glass cuvettes. A polycondensation reaction is carried out at 180 degrees Celsius and 0.3 torr for 1.5 hours. These reaction conditions are specific for CpHSA polyanhydride polymer synthesis. Using the automated deposition apparatus, the solvent and any components for encapsulation are deposited into the cuvettes containing the polymer library. Sonication can be administered to ensure completed dissolution of the polymer and uniform dispersal of the additional components. Then the dissolved polymer is transferred into a tube of non-solvent to fabricate the nanoparticle library by nanoprecipitation. The nanoparticles are recovered by vacuum drying or filtration and then transferred to a modified 96-well release plate. Finally, the protein-loaded library is incubated under physiological conditions, and protein release kinetics are evaluated using a fluorescence-based detection method. Ultimately, results can be obtained that show protein release kinetics as a function of polymer properties or release environment conditions.
The main advantage of this approach over other conventional one sample at a time techniques is that we're able to synthesize multiple samples simultaneously. These samples can then be screened in high throughput for protein polymer, cell polymer, or host polymer interactions. This then expedites the rational design and development of these biomaterials for drug delivery and tissue engineering.